Have you ever pondered about the importance of DNA sequencing in our lives? Imagine an intricate language of life concealed within our cells, waiting to be decoded. This is the enigma of DNA sequencing. It's like unlocking a vault of secrets that allows us to understand and even alter the function of genes. This remarkable technology reveals the gene's regulatory regions, the unseen architects that control gene expression. It exposes the genetic hotspots, those areas more susceptible to mutation, providing a window into the inner workings of our genetic makeup. Moreover, DNA sequencing offers us a time machine of sorts. By comparing DNA sequences, we can trace evolutionary relationships, providing a framework for a definitive classification of microorganisms, including viruses. It's like piecing together the jigsaw puzzle of life, one gene at a time. In essence, DNA sequencing is akin to deciphering the blueprints of life. So, how does DNA sequencing influence our daily lives and the world around us? Well, let's delve into the practical applications. Firstly, by comparing DNA sequences, we can unravel the complex tapestry of life, tracing evolutionary relationships that provide a definitive classification of microorganisms, including viruses. Imagine this like a gigantic family tree, mapping out the relationships between diverse forms of life. Further, DNA sequencing also enables us to identify conserved regions. Now, you might be wondering, what are these conserved regions? They are areas of the DNA that have remained unchanged over the course of evolution, serving as reliable markers. These regions are incredibly useful when it comes to developing specific hybridization probes to detect microorganisms in clinical samples. In essence, the power of DNA sequencing extends from the laboratory to our everyday lives. It's not just about understanding the blueprint of life. It's about applying this knowledge in practical ways, from tracing our evolutionary history to diagnosing diseases. DNA sequencing plays a crucial role. But what does the future hold for DNA sequencing? Well, let's dive in. As we advance in the realm of genetics, DNA sequencing is becoming increasingly rapid and affordable, transforming it from a specialized procedure to a common laboratory practice. This speed and cost-effectiveness are enabling us to determine microbial sequences, leading to more precise identification of microbes. Taking a closer look, we find that sequencing the 16S ribosomal subunit, a component of the prokaryotic ribosome, is a powerful tool for identifying specific bacteria. This method offers us a new level of precision in our fight against bacterial diseases. In the same vein, sequencing viruses isn't just about identifying them, it's also about distinguishing between different strains. This distinction can be crucial in understanding a virus's behavior, its response to treatments, and its potential mutations. As we continue to unlock the mysteries of DNA sequencing, we move closer to a future where we can fully understand and harness the power of our genes.